Jai Hind Bachcho. Today we are going to discuss another topic of chapter 5 Fundamental Unit of Life of class 9th. So in this we'll discuss today nucleus. It's an organelle present in cell. So let's start with it. So how can we see this nucleus? Yeah. So if we can temporary mount onion peel. What is this temporary mount means? We are making a temporary slide of onion peel. Temporary means we are not preserving anything. We can say make it and we will see the structure, right? And we'll just for that duration only it is available for us. Otherwise, if we'll observe it after 2-3 days, so after few hours everything will be spoiled in this because we have not preserved it. So there is one thing which is called permanent mount or permanent slides or temporary slides. So here we are talking about temporary slide or temporary mount of onion peel. So we all are aware of this onion bulb that we eat normally or use in veggies as veggie. So if we cut it, right, and take single part of that or a single leaf of it, right, so and we peel it from inside, then we'll get this transparent structure, right? And if we mount this on a slide, right, and put a glycerin and put a cover slip, right, or we can add water also, we can add water and then put a cover slip, right? So, and if we see it in the microscope, if we'll not stain it, right, then we will not able to see that in that slide, and we will not distinguish different parts of a cell, right? But if we'll stain it, staining means we'll put some chemicals, like we can stain it with saffronin, we can stain with iodine, or we can stain with the methyl blue. So these are the stains. They give a color to the cell, and that color, different part of the cell will color differently means somewhere it will be a dark color and somewhere it will be the light color depending upon the chemical composition of that particular organelles or part of the cell, right? So if we stain with saffronin, so it is little red color chemical. So we'll see the cell in a microscope, compound microscope like this, and in that we'll see here there's a light pink area, and at the side there's a dark pink structure, dot like structure, that will be dark pink. So this dark pink structure is actually nucleus, right? So here we are studying the onion peel, so it's a plant cell. So structure, the dark pink structure, that is nucleus, we see at a side because the main part is occupied by vacuole in plant cells. Clear? Same way we can study animal cells also. So how we'll study, like cheek cells. So from the inner lining of our cheek we can scrap from with the help of some scraper, right? Or we can use toothpick also, and we'll put that on a slide and put a cover slip and stain it, put on in this on the slide and put a stain, that is saffronin or any other. So mainly we use saffronin in labs. So here also the cell will color differently, like this center will become dark color, a dot like structure in the center, and outside will be a light color, right? So in the center, which is dark color dot like structure, is nucleus. Clear? So in animal cell the nucleus occupy the central position, and in plant cell it is at the side. Okay? Now if we talk about the little bit detail about this nucleus, so we can see structure like this. See, this is a cell. We have drawn a rough sketch of a cell. So this is the cell membrane. This area is cytoplasm. Then the center is a nucleus. So nucleus, it is surrounded by membrane. Okay? And the membrane is double layer. It's not a single layer, it's a double layer. So you can see here. And there are pores. Okay? So there are pores. From the material can move in and out of the nucleus. Okay? The nuclear membrane, these are the pores. And in the nucleus, there is genetic material. Okay? So when cell is not dividing, that time we call this genetic material as chromatin material. Clear? And what this chromatin material is made up of? This is basically DNA and proteins. Right? So number of proteins are there and DNA. So this is the basic structure of a nucleus. Now what is the full form of this DNA? Is the deoxyribonucleic acid. Right? So how this chromatin material is organized? Like if we say what is the why protein, DNA, we are talking these terms. So basically what is this? This chromatin material. Right? Chromatin material when cell is not dividing. At that time we call it a chromatin material. Right? When cell is not dividing. So what is this? Actually the DNA, how it is organized? The DNA is actually very long thread-like structure. This is double helical structure that we have discussed. You will study this in your higher classes. It's like this helical. And it's very long. Right? So it has to be organized. Right? It has to be packed in that small nucleus. So how it is packed? It's like these proteins, they are the ball-like structure. And around them it is coiled. So DNA is coiled around proteins. So this is the basic structure. This way the DNA is packed in the nucleus. Otherwise it will be very difficult to stay because the nucleus is very long. And how many number of this chromatin material in a cell? If we talk about this animal cell or if human being cell. So we have, like this is center nucleus. So number of chromosomes are, we have, or chromatin material, if you talk about this, we have 23 pairs or 46 in number. So we have total, like this DNA, we have total 46 DNA. Right? And it is organized same way. DNA is coiled around protein. So this is called chromatin material. So don't be confused with this word. So this is just DNA and protein that we call the chromatin material. And what is this DNA contain? Right? This contain information for constructing and organizing cell. Right? So what will be the cell, what will be its chemical composition, how it will function, how it will be organized, everything will be decided by DNA. So all the information is there in the DNA. So this cell will be going to be a mango plant cell or a papaya cell or a human being or a pea plant, anything, cat, dog, anything. So it will be, every information is there in this DNA. Clear? How this nucleus will be when cell is not dividing. And so when cell is not dividing, so what is there? Same way, there is a membrane is there. This is double layer nuclear membrane. This is cell membrane. This is double layer nuclear membrane, nuclear pores, and this entangled mass of thread like structure, that is chromatin material. What is this chromatin material? Again the DNA protein. And how is DNA is organized? It is coiled around those proteins, same way that we discussed here. So this way this DNA and protein will remain, and we call that as a chromatin material. When cell is not dividing, it's just a thread-like structure, and it is not visible in the microscope. If you see in the microscope, you can't see it clearly in undividing cell. Now what happens when cell divides? So when cell is about to divide, so what happens? The same chromatin material. See, this is cell membrane. This is nuclear. Roughly, we have drawn this nuclear membrane, and this chromatin material itself, it thickens. Right. So this DNA, I told you, it's a long thread-like structure called but again it thickens more, right? And that structure we call chromosomes. There's nothing extra. Same thing is there, same material, same DNA, same protein, but that is thickened. That we call the chromosomes when cell divide. Right? And more about this you will study in your higher classes. See, when cell divides, the no nuclear membrane, this chromosome, they become more thickened. We can see this type of structure in the chromosome in the dividing cell. Chromosomes of dividing cell. So these are chromosomes. So don't get confused with the word chromatin material or chromosomes. Right? Here also DNA is there. Yes, there is a difference in the number of DNA, because cell is dividing, naturally the DNA will divide. That will be there. That is the scope of your higher classes, right? But it's same DNA and protein. It thickens, that we call chromosomes. Now, this is the DNA that we, if you remove the protein, if you only talk about the DNA, here we are not taking into consideration of protein. Otherwise, DNA in the cell, if it is protein, it is in this form, DNA coiled around proteins. Right? This is the way of packing of DNA by nature, you can say. See, if we talk about the single DNA contain information for number of characters. We know that you are all familiar with, and you studied in lower classes, that DNA decide our fate. Everybody's DNA is different. So how it is, everybody's DNA is different, and how it works actually. So if we see, this is a long DNA, double helical structure, like structure you will see in study in your higher classes. But just for now, this is the DNA, right? So here we are talking about the single DNA, right? So in different plants or different animals, the number of DNA will vary. Like in human beings, there are 46 DNAs or 46 chromosomes. 23 pairs we are talking about, 46. Like in pea plants, there are 7 pairs of DNA or 14 DNAs. 7 pairs, 14 DNAs. So here we are talking about like single DNA. So these all DNA, there's a 14 DNA or 46 DNA, or some may be having 23 DNA, some may be having 20 DNA. So they all control the character of that particular organism. And how? So like this is a DNA. So suppose from here to here, this length of DNA carries information for height of that particular organism, right? So, and suppose from here to here, it carries information for the shape of nose, right? From here to here, this length of DNA, it carries information about color of skin, and so on. There are a number of characters. So we have taken few. So what is this length of the DNA called? Which carries the length of the DNA, which carries information for a particular character. So height, shape of nose, color of skin. So we call this genes. Clear? So genes are functional segments of DNA are called genes. So functional segments of DNA are called genes. I hope it's clear. Now, this role of this nucleus. What is this role? So, central role in cellular reproduction. We know that first nucleus divide, chromosomes they divide. Nucleus divide, then only cell divides. So, its role in the reproduction. Secondly, and it will determines the way the cell will develop. Right? The cell will develop into what type of organism, right? So from one cell only every individual is arise. So this cell will develop into a mango tree, or this cell will develop into a cat, or this cell will develop into a dog, or anything of a human being. That will determine by this nucleus. So determine the way the cell will develop, and what form it will exhibit at maturity. So what will be the cell will be at maturity. That every information is there in the nucleus. And how it is doing by, see, and how it is doing, like information is there, but that is directing the chemical activities of the cell. See, so information is there for the, like in the DNA, this information from here to here. Suppose this part is control the color of the skin. Color of skin. How dark will be skin, will be fair, or there will be number of variation in the color of the skin in human being if you talk about. So information is there. So this will control the chemical reaction of cell. Chemical reaction means how is the color of the skin will define? The quantity of melanin pigment, number of melanin. If melanin is less, then color will be fair. If melanin is more, color will be dark. Right? So this will define where this melanin will be produced. The cell will produce the less melanin, or will be produce more melanin. Right? So their information is there, and it is controlling that chemical process. From here something will be formed, that chemical will form. So who will control this all reaction is this. So this is only we are talking about. Like, first of all, the role of nucleus is in reproduction. The cell divides from 1 to 2, 2 to 4, right? And secondly, it determines the way cell will develop, right? In what form it will take, and what form it will exit at maturity. Okay? How? By directing the chemical activities of the cell. So I hope it is clear to you. Right. And if we further talk about this nucleus, so there are cells where there's nuclear membrane is there. We have talked about this is cell membrane, and this is double layer nuclear membrane. Nuclear pore and center this chromatin material. So cells where this nuclear membrane is present is called eukaryotes, or eukaryotic cell organisms are called eukaryotes. But there are cells where this nuclear membrane is absent. Like this is cell membrane, and this is genetic material. Right? So here that region is not defined. That nuclear region is not defined. It is not surrounded by membrane, right? Or poorly defined nuclear region. So this we call nucleoid. We will not call nucleus. This we call nucleoid, right? Nucleoid. And such type of organism we call prokaryotes. Right? Pro means primitive, and karyotes means karyon. It derives from word karyon and means nucleus. So it's a primitive nucleus. So this type of structure or cells we will see in bacteria, where there is no cell membrane. So bacteria are basically prokaryotes. If we little bit study more about this prokaryotes. So in this, these prokaryotes, right, they lack most of the other cell organelles, cytoplasmic organelles, like those who are membrane bound organelles are absent, right? Those who are present in the eukaryotic but absent in prokaryotes. So no membrane bound organelles are present in prokaryotes. I will, like mitochondria, chloroplast, they all are absent. Even the nucleus is not surrounded by membrane. We don't call it nucleus, we call nucleoid, right? So what the basic structure of a bacteria prokaryotes? They have cell membrane, they have cell wall, right? Then this is the genetic material and ribosome and some more other organelles that we'll study in higher classes. Not organelles, some other chemicals or vesicles or vacuoles, right? See, and there are prokaryotes, those who are photosynthetic. They make their own food, right? They are parasitic also. They are bacteria or prokaryotes are parasitic also. They are photosynthetic also. So how they will prepare their food? So they have contain chlorophyll, but that is not present in chloroplast. Normally in eukaryotes, we know that chlorophyll is present in chloroplast, but here it is not present in chloroplast. Then where it is present? Like if this is the cell wall, then the membrane they have infoldings. These called the vesicles. The membrane they have infoldings. In that, the pigment are present for photosynthesis. This is called vesicles. So no membrane bound chloroplast is there. Plastids are absent. So these are the vesicles where these nucleoids are, these pigments for photosynthesis are present, right? So this is all about nucleus. So another organelles we'll study in next video. Thank you, students.